Hi, my name is Troy Blanchette, and this is my senior project presentation of a study of the Millis microbiome. A microbiome is the collection of all bacterial and microscopic factors in a community. And in this presentation, I will outline exactly how I analyze our microbiome in our community. I got this project idea because through STEAM Scholars, I need to do a job shadow my junior year of high school. And I chose to job shadow my aunt, Jennifer Jankovic, at her lab at the York Main Hospital. And what she does is when a doctor takes samples from a patient to be able to diagnose them, they send it to her lab where she grows and identifies the bacteria they have to give a diagnosis. And from that, I asked myself, what bacteria could I find in Millis? So that's how I came up with the idea of a microbiome study. And I knew that this would entail the collection, growth, and identification of bacteria. So the mentor I chose for this was obviously Jennifer Jankovic, who is my aunt. She has a degree in medical laboratory science from the University of New Hampshire, and she works at the York Main Hospital. I also had easy communication with her throughout this entire process because of our close relationship, and she is very knowledgeable on the subject as she is the head of this lab, and she has been doing this for many, many years. The research statement I made for this was the collection, growth, and identification of bacteria from a common space. Collection, growth, and identification being the three main steps in this process. I hypothesized that the school environment is a relatively clean space, thus few pathogens will be found on the school library desk. A pathogen is any type of bacteria that could get a human sick. The first part of this was collection. I chose to collect from the school library front desk. I chose this location because it is a place that is commonly touched by both middle and high school students. And for this, I had to make a very specific swabbing procedure. I chose to do 6 different sections, each 4-inch by 6-inch templates. As you can see at the bottom here, that's what the templates looked like as they were down on the desk. I had to use sterile swabs, which I got from the biology room from Mr. A. And this specific swabbing procedure used wet then dry swabs. So first I would wet a swab with distilled water, and I would go over the section outlined by the template in a diagonal pattern. What this would do was the bacteria that sits on the top of the desk, as the water goes onto the desk, it picks up all the bacteria, so the bacteria ends up swimming in that water. I then go in again with a dry swab and picking up all that bacteria-infested water. I then had to transfer this onto agar. Agar is a type of gel that sits in a petri dish, and this is where a bacteria grows off of. The specific way I had to put this on used a procedure with quadrants. As you can see at the bottom right there, you can see that in each quadrant I did a diagonal pattern, and that's how I had to place it onto it. Also on the top, you can see me putting it on. The second part of this project was growth. There are many different agar types I used. There are 2 main agar types. There is non-nutrient and nutrient agar. Nutrient agar has some sort of nutrient in it that the bacteria can use to grow, while non-nutrient agar is basically just a gel and it doesn't have that nutrient for it to use. Nutrient agar is the generic type. However, I did use generic nutrient, but I also used sheep's blood and chocolate or chalk agar, which are 2 specialized types of that. I also had to do incubation. The incubator I used was found in the biology room, and I got permission from Mr. A to use that. And I had to incubate it for 24 to 48 hours for each collection at 35 to 37 degrees Celsius. You can see down there that is the incubator, and at the top there you can see that is chalk agar, and you can see all those little dots. Those are all colonies of bacteria. You can also note that some of them have different morphologies, as some are different colors and different shapes, and that's how I partially use the distinction of that to identify the bacteria. For the identification part, I did identification myself, and then I also took different samples to a lab to be able to identify it much more thoroughly. For my self-identification, I used a very lengthy process called gram staining. What this is, it's the use of chemicals such as crystal violet, iodine, and safranin to be able to distinguish between the 2 major types of bacteria. There is gram-positive and gram-negative bacteria. The main difference between these 2 is that gram-positive bacteria has a much thicker cell wall, and by distinguishing these 2, that's how I identify the bacteria because I know which group it belongs to. I also had to observe these through a microscope. So after I do the gram staining, gram-positive bacteria under a microscope will appear violet, and this is because the crystal violet adheres to the cell wall, while gram-negative bacteria, the iodine and safranin adhere to the cell wall, giving it a pink color. At the top right there, you can see this is me putting on crystal violet onto the slides. And at the bottom, these are the 4 sections that I gram stained, and that's them after the gram staining. I then visited my mentor's lab at the York Main Hospital, and I used a big machine called the MALDI identification machine, and you can see it at the top there. What this machine does is it quite literally shoots a laser down at a petri dish with bacteria on it, which explodes it upwards in a projectile way, and through the colony morphology and the actual trajectory of the bacteria, it is able to exactly determine what type of bacteria it is. So for my data and results, first I had trial results. I did 4 trials of collection and growth before I even considered and started the identification. This is because I had to consistently be able to grow the bacteria to know when I could start identifying it. For my first trial, I used the non-nutrient agar, which yielded no bacterial growth, and this was most likely because it didn't have any nutrients for the bacteria to grow off of. Then for the next 3, I used nutrient agar, chalk agar, and then sheep's blood agar. And all of these yielded bacterial growth. As you can see at the bottom, that is chalk agar, and it yielded a lot of growth. It is also notable that the chalk agar and the sheep's blood agar typically grew more colonies of bacteria than the generic nutrient agar. So this means 3 of the 4 initial trials grew bacteria. And after that 4th trial, I was confident that I could begin my identification process. So what I did first for identification were my individual identification, which was the gram staining and the observation under a microscope. And out of the 4 slides that I observed, I was able to identify 2 of them. The first one is Lactobacillus rhamnosus GG, which you can see at the bottom right. That is what it looks like under a microscope. It is a type of gram-positive bacteria that is found commonly in yogurt and other dairy products. It is very healthy for the human gut, and it makes sense why it would be on a school library desk, as if someone ate yogurt and then touched the desk. I also found on slide 3 Enterococcus faecalis, which you can see at the top under a microscope. That is another gram-positive bacteria that is found in the human gastrointestinal tract and in human feces. So what this might reflect is that someone did not wash their hands after using the bathroom and then touching the library desk. Other data and results are, this is a complete list of all the bacteria I found from the MALDI identification machine. It is a complete list of many different types. Beside each one, you can see a number and a color. So I will explain how the MALDI identifies and what it gives me in that. The number represents how many colonies it found. So there were 4 different types of colonies out of the sections, because there were 4 sections I gave. And so in all 4, Staphylococcus hominis was found. And then there is a color next to that, and that is a confidence reading of the MALDI identification machine. Red means low confidence, yellow is medium confidence, and then green is a high confidence reading. So out of all of these, the most notable is Staphylococcus hominis because it was found on all 4 with all of them having a green or high confidence identification. Staphylococcus hominis is a bacteria found on all human and animal skin, and it contributes to body odor, so it really was not surprising that I found it there. And on the right, you can see 2 of the sections that I used on my identification here. So what I concluded about this was that the middle school library is a clean space, as there was rare bacterial growth and no pathogenic bacteria found. So although it may have seemed like the petri dishes were teeming with bacterial life, in reality that was considered rare bacterial growth because heavy bacterial growth, almost the entire agar plate would have been covered in bacteria, so it would have been white or yellow. So it was actually very good that it was rare bacterial growth because that reflects that there wasn't that much bacteria on that school library desk. Also, out of both my individual identification results and the MALDI machine, no pathogenic bacteria was found, so that was very good, meaning in normal amounts, nothing could get someone sick. My individual identification results were not supported because I did not find the exact same bacteria when I used the MALDI. However, they're still likely accurate because I did find similar species of bacteria and I did find other species in the same genus. Some error that occurred was that the MALDI machine gives analysis might have some error when the colonies mix. That's when we have the red or yellow, the low confidence reading, is when 2 colonies mix, the MALDI machine gets a little confused. However, when it is just by itself and it gives a green reading, that has 100% accuracy. The next steps I would take with this, if I had more time, I would ask, are all spaces in Mills High School as clean? I only identified what I found on a school library desk. So I could do collecting, growing, and identifying bacteria from other commonly used spaces. What these might entail would be doorknobs or light switches. Some challenges I found was that in some of the Petri dishes, there was fungal contamination. And when there is fungal contamination, if you were to release it, there would actually be spores and it would become airborne. The only place I would be able to open that would be under a hood, because hoods have a suction. However, the only hood we have in Millis High School is found in the chemistry room, and Ms. Copese clarified that it is not strong enough for that, so those I had to just throw away in the hazardous. I also had some challenges with the incubator at first because it took hours for it to reach the temperature I wanted of 35 to 37 degrees Celsius. And it didn't reach that at first, so I had to plan a few hours ahead to set it. I also need more tests for my individual identification because I only identified 2 out of the 4 slides. If I had other tests, such as lactose fermenting tests, I might have been able to identify those. For the vision of a graduate, I use communication a lot in my permissions from both Mr. Adicki and Mr. Engler, and I had interviews and conversations with my mentor. I had to collaborate a lot with Mrs. Boise and Mr. Carter on use of the library. I used critical thinking a lot in both my research, my making of the procedures that I had to use, and in my problem solving of the errors that I found. Also, character was used here when I had to analyze an environment that we all use, and I had to work well with others. This is my works consulted.